Okay, so now that we have some basic understanding of the structure of DNA, we go right into the replication process. And that's really what Watson and Crick did as well. Their elucidation of the actual structure led them to hypothesize on how it would be replicated because it made sense structurally, sort of that process, how it would work given the specific structure that they found out. So what we're going to go over is a very simplified version, although you might still find it complicated. It's very simplified by comparison to the actual version. A bunch of pieces I'm not going to include because it just adds to extra level of knowledge you have to have coming into it, and certain details that you probably just aren't going to need to understand the big picture of the process. So we're going to be focused on big picture. And if there's something that is needed, it's a fine detail to understand that, then we're going to include it. But if it isn't really necessary, then that's not going to be included here. I'll have other lectures that go into more detail on all the enzymes and proteins and everything else if you happen to be interested in that, but you won't be held responsible for that. This is kind of the core version that we're focusing on here. So keep in mind, DNA is double-stranded. It is a double-stranded helix. So the two strands spin around and wind around one another, right? That the strands are anti-parallel to one another, right? So that, say this one, if there's a phosphate at the end of here, there's an OH there, and at the other end of this one is an OH, and this one has a phosphate. So they're set up like that. We're going to be, or I'm going to be, drawing these structures here kind of linear, kind of straight like this. And it's not exactly how it is, but it's just going to be a lot easier for you to see it and for me to actually draw it. And I'll add in a couple little details. So the two strands of DNA, let's say we have A, and I'm going to do a couple little things here like this, like C and G, just to kind of, so we know that the strands are upside down. Do T here and A there, like that. So this one had the phosphate there. This one would have a phosphate here. This one had an OH there. That one would have an OH here. Okay. And remember, the two strands are held together with hydrogen bonds, right? So we got hydrogen bonds here. There are two bonds between, so A's and T's have two hydrogen bonds. G's and C's actually have three hydrogen bonds. And that's what's holding these strands together. So what happens overall in the process, which we call semi-conservative replication? Semi-conservative because totally conservative would be this is the original, say, DNA, and it would stay together always and never be pulled apart or broken down in any way. And so this is going to keep it a single strand intact, but we're going to separate the strands. And that's the idea. So in DNA replication, very generically, one of the first things that has to happen is that the strands get pulled apart, and the whole entire thing doesn't get pulled apart, just get the pieces get pulled apart. So we'll get into that a little bit later. So the two strands are separated in some region. The T here, and then we got here the T, G, and A here. Yeah, that's why that looks wrong. I did it right side up. It's supposed to be upside down. Okay, so without drawing any other stuff, we'll just kind of leave that here. I'm actually sorry because I'm going to draw extra stuff here. And now I realize I need more room. I'm going to just redraw that part anyway. Okay, so T, G, A. Okay. So each strand, now a single strand, so this is, see, we might refer to this as DS, or double-stranded DNA. This is now SS, or single-stranded DNA. It's not totally all by itself. Again, it's still kind of connected with the rest of it, but this is just, we're talking about, say, let's say, for example, in this area here, do this, we're pulling the DNA apart, in a way. I think I used this one here. Okay, so there's a section here that isn't, there's terms to talk about this, that isn't connected. So the bonds have been broken, they've been pulled apart. Okay, so it's not the whole thing, it happens in a little section, but that's what we're focused on right now. It's like a little section of it. So for the concept, we pull the strands of DNA apart. While they're apart, each one can be a template, meaning that if you were going to build new DNA along this, what would it look like? Okay. So it would be a new strand, and you'd have to, well, based on the rules, you'd have to put a T there with the A, and you'd have to put a G here with that C, you'd have to put an A here with that T, and they would go together, bond, bond, bond. This one, you would build a strand like this, and this one would have an A, C, T, like that, bond, 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 like that, and then you would get two new double-stranded DNAs. And that's the overall idea of DNA replication, that one strand gets pulled apart, each side serves as a template, and then enzymes can read the information on what we call the template strand, and then can use that information to make new DNA. And then in the end, you have these two new double-stranded DNAs, and DNA's been replicated. If it were just that simple, it would be, I'm sure, very nice for you, but it's not that simple, and there's problems with some of the things I said. Nothing incorrect, just we're overlooking stuff. We're overlooking certain issues, right? So issues like the way that DNA is built. So how do you actually build DNA? Does it matter? Is there a direction? Are there any rules or requirements? So first off, who builds DNA? So new DNA is made by an enzyme called DNA polymerase. Makes sense, okay? So it's the name. DNA, well, it makes DNA. Polymer, and ASE. All enzymes end in ASE. So it's an enzyme who makes a polymer of DNA, so it's going to join together individual nucleotides, like we drew in the last lecture. An A, and then a G, and then an A, and then a T, and then a C, or whatever it happens to be. It's going to make the new strand in order. How does it know which ones to add? It's going to be looking at the template to say, oh, it's going to say, hey, I'm an enzyme. I'm working here to build a new piece of DNA. Who should be the first one? Oh, thymine, because there's an adenine there. So I'm going to move on. Who's the second one? Well, here's a C over here, so it's got to be guanine. It's got to be the second. I'm going to keep moving. I'm going to add another one. Who should be here? Oh, thymine's over there, so that means adenine. So it knows which one to put because it's reading the information from the template strand, like that. So the enzyme's working along. But there's a certain direction, there's certain rules. So first off, the DNA polymerase can only read a template in a certain direction. It can't read it in any direction, right? So we have our 5' directions and 3' based on the phosphate OHs, right? So the DNA polymerase can only read DNA 3' to 5'. So that's OH to phosphate directionality. So that means it can read, if it was going this way, it could read, say, this strand in this direction, but it can read this strand in this direction. So that means if one of these was going one way, that means it would actually have to be reading this like that. So that means it's building this one, along it. But remember, the strands are anti-parallel, so it can only build DNA 5' to 3'. Okay, so that's now phosphate to OH. So the other strand has to be opposite, right? So that makes sense, should make sense. So these are some rules. So starting off here. So this first rule, the rule about it can only read 3 to 5, right? And it can only build DNA 5 to 3. First two important rules. The next rule, also very important here, it cannot build from scratch. From scratch, okay. Well, what does that mean? So kind of what I did here was building from scratch. I just kind of, there was nothing there, right? There was nothing here at all. And I just wrote down T, and then I added to that, say with the G and then the A. But the key was the first one that I added there. I added it with nothing before it or in front of it. And you would say, well, of course, how should there be something in front of it? If I just pulled these two strands apart, what's attached to this strand? Nothing, because it used to just be attached to that strand. So there's nothing there. There's nothing to build off of or attach this to, except this is one of our big problems. The only way this enzyme works is if it's making something that already exists longer. But nothing exists attached to that, right? So it seems like a paradox. How can it do something that it can't, by definition, do? And that's because there's something we're leaving out, right? There's something else here. So there are other proteins and enzymes all working together, right? So I'm going to list a whole bunch of them for you here. You don't have to know all of them. I'm just saying it, not to say, oh, I know all the names. I'm just saying it because, just to give you an idea, there's a lot of stuff happening, okay? So there's an enzyme called helicase, which we are going to talk about, that's actually breaking the hydrogen bonds and pulling them apart. There's things called single-strand binding proteins that are attaching to these temporarily to keep them single strands. There's something called topoisomerase, because as these things are coiled, the DNA is being pulled apart. It actually is like getting twisted tighter down here. And that's going to actually tie knots in it, and it's going to like mess the whole process up and destroy it. So there's an enzyme to kind of fix that. And then we get to the enzyme that we need to talk about right now called primase. So primase is an enzyme that's going to create little things called primers. Okay, so it's going to build from a primer. So because the DNA polymerase enzyme can't build from scratch and just start to assemble these things, it has to already have something there. We have to put something there. Your cell has to put something there. So the cell uses an enzyme called primase to build a little thing called a primer. And the primer is a small piece of RNA. Short piece of RNA. That's what this is here. I'm doing green here. There's a little bit of RNA attached to the DNA. Like the key thing is here, what's its purpose? Our point is it's to provide a free OH or hydroxyl group so that the new DNA, it can only be built 5 to 3. So the next one, so this would be the 5 over here. So it can add the next nucleotide. So here, and then the next one, and then the next one, and the next one, and the next one. So it can kind of be building from that primer. And the same thing here, it's going to then build. It's going to be building that way. Because it can only build 3' to 5', only build 5' to 3', and it's only reading the template 3 to 5. So that's what's happening right now. There's another problem, and that's what you're going to see. You're going to see some pictures of that drawn better, probably, than this, that are associated with our book. And if you just look it up online in general, you'll see those pictures. But there's another problem, right, with those pictures. Okay. I'm going to try to show it to you here in another drawing. Okay. So if we have, I'll use the pinks as the templates, or the originals. Okay, so this is the original DNA that's being like pulled apart. And then we have our RNA primer. Let's say a primer here. And there's I'll put a little primer here, like that. And then we have our new DNA being built. We're saying this new DNA is being built here. Now you'll see a picture looks a lot like this, your book and a bunch of other places. It's being built by the enzyme, the DNA polymerase. I don't know if I color-coded that or anything yet. No, I'll use this blue one. The DNA polymerase is doing this. Again, you'll see a lot of pictures that kind of look like this. You say, all right, that's fine. I kind of get it. It makes sense. Hopefully that makes sense at this point. But the catch is this. And the place where the other drawings are kind of wrong or misleading is that, and mine currently isn't, but I'm going to fix it, is that these DNA polymerases, these two enzymes that are working on these opposite strands that are reading the DNA 3 to 5 and making the new DNA 5 to 3 are physically connected to one another, and they're both moving in the same direction. They're both moving in one of these directions. So they're both moving this way, they're both moving that way, but they're moving in one direction together at the same time. So now, once again, we have a problem, just like before. The paradox of problems, like, but that violates these rules. That doesn't seem to make any sense for part of it, at least. So this one, the direction of replication and the direction of movement are the same. So it's reading in this direction, it's making the new DNA in this direction. It can continue in this direction without any problem from that one little primer. So not an issue, not a big deal. Here, we have a problem. Because we're saying we're dragging this now in the opposite direction of where it's supposed to be making. It's supposed to be making DNA this way, but we're pulling it downward, right, in the opposite direction. So this one is going to be, in a way, like attached constantly. This one is actually going to be like jumping. It's going to attach and then detach, and then attach and then detach, and attach and then detach. It's going to be what's called discontinuous. So the terms here would be continuous synthesis. So for one of the strands of DNA, the one that we're going to call the leading strand, the direction of movement, the direction that the replication process is working, follows the DNA, one of the strands, that's going 3' to 5'. And so it all works together. The other strand, since it's anti-parallel, means it's moving opposite the way it's supposed to read. So it has to read it this way and make the new DNA, but it's being pulled the opposite way. So along this strand, we're going to have this stop replication, which is also called discontinuous synthesis. And the other term here is that this strand here would be referred to as the lagging strand. All right, so when we talk about leading and lagging strand, we're talking about the template strands, the original strands, right, that are being used while new DNA is being built. But the new DNA is being built by an enzyme called DNA polymerase. There's one enzyme for each strand. Both of them follow the same rules. They can only read the DNA one way, just like you would read a sentence. Starting here, you wouldn't start backwards and go the other way. You start the first letter and go in this direction. Usually things also, we often read from top down. You know, we don't usually read from the middle down and go back up the top. I mean, I'll skip around when I'm talking about things sometimes with my outline, but generally there's a way. You know, we kind of start at the top and then we read across and we work our way down. That's the way generally that we tend to read things, and these enzymes have certain rules as well, and they read in a certain specific way by their rules, okay? And they can't break them or change them. So we have this situation where for one of the strands, it's not a problem. You learn the basics and the basic rules, and that's exactly how it works. It starts from one little primer of RNA that's used to build off of, and then it just builds continuously, non-stop synthesis of new DNA. In the end, we'll have a new double-stranded DNA, the new strand and then the original strand. On the other strand, we'll end up with the same thing. But in order to do that, because the whole mechanism is being pulled in the opposite direction of where it can read, it's going to have to kind of jump forward, read and then do this little bit. And then often it can only do a little bit. It can't even do the whole thing. So there's going to have to be like another primer. And then it's going to have to do, this bit, here. And then there's going to need to be, another primer here. So it's going to have to jump, and then do this bit. I mean, they'd be doing them in order. This one first, then this one second, then this one third, like that. It's the order, not skipping around how I did. But that's what they'd be doing. Start here, make this, stop, jump ahead. Then they'd make this section. Then they would stop, because they already made this piece, or there's a primer there. Then they'd jump ahead. Then they'd make this piece. And they'd keep stop, and then they'd make it, because there's already something there. So they just keep starting and stopping in a discontinuous synthesis process. You're still going to get new double-stranded DNA. You're just going to have a lot more primers, right? Because you have to keep, every time you start, you need new primer, new primer, new primer. Okay? So there's more to it than this. There's a lot of other things happening, but these are the core things to really get the big picture of the process. So semi-conservative, we're going to end up with 2 new double-stranded DNAs in the end. We got the enzyme doing it. We have some basic rules of how it's done. One thing we're going to still add in here, and then I'll talk about a few other things with the terms over there, is that in the end, we have these primers. They shouldn't be there. So it's kind of like DNA RNA hybrid. So there will be enzymes that come in the end that do what's called proofreading. So proofreading enzymes will actually look and try to compare, is A with a T? Is G with a C? Is T with a T? Like, whoa, T with a T, that's not right. It should be an A there. So what this proofreading enzyme can do is look at all the base pairs, and if it finds ones that are wrong, it'll determine which was the original strand, the template, and which one's the new one. It's a sort of chemical labeling. And it will then be able to cut out or remove the nucleotide from the new one, because that's the one that would be wrong, and then it'll replace it with the correct nucleotide. When this proofreading enzyme gets to the primers, it's going to say, this is not even DNA. This is RNA. So we are going to cut this out. So the enzyme, the proofreading enzyme, will cut out those primers and then add in DNA nucleotides. So it'll kind of come back, cut out this primer, add in DNA nucleotides, and so on. There's some other issues here. There's going to be a gap that has to be sealed by another enzyme called ligase, which comes in later, which we're going to kind of gloss over here. It's adding a little too much because of all the other stuff we still need to cover. But in the end, it'll remove the primers. They don't stay there. They all go away, and then we end up with DNA. And that'll happen on the leading strand for just the few primers it has, but happen a lot for the lagging strand. One other thing to kind of talk about here. Where replication begins. So let's say replication starts right here. And so that helicase pulls the DNA apart, right, in this region. What will happen is that DNA replication will occur in this direction and this direction. So I didn't just say that direction because it was specific. It's both. Okay. So it's what's called bidirectional replication. That means from the origin, this, an origin of replication, the place where the DNA starts being pulled apart, it's typically not the end, right? Like this. It's actually somewhere within the DNA. It pulls the DNA apart, and then it works in both directions, right, from that point, because our chromosomes are so large there are often multiple origins. So there might be one here and another one here and another one someplace else and another one someplace else. And they're all kind of like working together. Eventually they're going to converge and all hit one another. And then these other enzymes, like the proofreading enzymes and that ligase enzyme that I mentioned, we're going to have to like splice everything together, put it all so just they're continuous pieces, because there's going to be fragments essentially right after the process is over. The other thing that's so the whole so that's called an origin. That's one term. The next thing is this whole area here, this whole area where the DNA is being pulled apart. That is usually referred to as a replication bubble. This is kind of like, if you look at it, it's kind of like being pulled apart here. So that's the replication bubble. That was supposed to be an R. Replication bubble. And then if we just look at a piece of it, so like we just look at one end of the replication bubble, that would be called a replication fork. So essentially what's happening here is happening in both directions at the same time as well. All the same enzymes doing all the same things, following all the same rules, but just in both directions. So half of that's called a fork. The whole entire area being pulled apart is called the replication bubble, and that is beginning at some place on the DNA called an origin. Bacteria can have usually just say one origin on their one chromosome. Other organisms that have more chromosomes and bigger chromosomes typically have more origins of replication, multiples. Some bacteria can have multiple origins as well, but many of them just have one. So I think that is the big picture of everything, and we'll return to this again as we start into now mitosis or cell division. So kind of keep in mind, there's a point to this, right? The point of this process is that a cell wants to divide into two new cells. Each of these cells, or the original cell, had a nucleus, and inside the nucleus was DNA. So before you can get two new cells, each with their own nucleus and their own DNA that's exactly the same as each other and as the original, somehow we have to duplicate everything. We got to double every single thing in the cell, starting off here with the DNA. We got to double the DNA before we can divide. So our next process is to talk about cell division. And that's kind of what this leads into. So we'll kind of, this isn't the end of it. We'll end this as we start to talk about replication, because what we'll have are like two chromosomes that are exact copies of one another. So that's kind of still the end of this process.